main primary focus of what you normally hear about. My talk actually, or my research goals are more about providing or learning about a better understanding for pecan nut development as a whole process. So I have sort of two interrelated topics today. One is, in general, what happens? How do you build a pecan nut? And you know, Charlie mentioned we got a little bit of grant money. That was partly because I had help. I had help from Charlie and some other USDA scientists, and that's kind of what it takes. I also was able to get a little help from Joe in a letter, and that's the kind of cooperation that actually made that application succeed. And I hope we can build off of that, because what we have right now is, I'll tell you a little bit about it today, it's just a good beginning, but I think there's a lot more we can learn from this. And while these results aren't going to be immediately applicable to the growers as a whole right now, I think a few years down the road, the things that we're going to learn will be helpful. And as my title suggests, part of my research responsibility at USDA is to research food allergens, and in particular nut allergens. So before I start talking about the pecan development project and pecan allergens, I'd like to give you a little bit of background on food allergens. I'm lucky, I have 2 daughters and a new little boy. None of them seem to have these food allergies, but we have neighbors and family, friends and family who actually do have this problem. And you'll hear some of the statistics about this, and I just want to give you my point of view, is that in some instances these statistics may be slightly exaggerated, you know, depending on how they get them. But in one sense, if you have a child with the allergies, it doesn't matter what the numbers say, it's just something you got to be aware of all the time. But it's not just nuts and peanuts that cause these allergies. There's a group of 8 foods called the Big 8, shown here. These 8 foods account for probably 80% of the food allergy that's reported. Milk, soy, eggs, wheat, shellfish, fish, tree nuts, and peanuts. A lot of these are actually outgrown. For example, milk, egg allergies start out in children, but as they enter into adulthood, roughly 80% of those allergies will be outgrown. If you compare that to peanuts or tree nuts, and it's a much lower number. Peanut allergies outgrow maybe 20% of kids, whereas tree nuts, it's even lower. It's maybe only 10% of allergies are outgrown. The impact on this, you know, the medical costs, about $500 million annually. That was back in 2007. I think that study was done. Cases of anaphylaxis are most often caused, the ER visits are most often due to accidental food exposures when someone has a food allergy. And again, these numbers, you know, 5% of U.S. children under 18 suffer from food allergies. That's all foods. But that's also, in some of these studies are from self-reported cases. So it's not actual diagnosis. It's a call to population centers to determine the numbers. And so there may be some you know, variations in that between what's really out there and what they report. But if you have someone you know or someone in your family member with an allergy, it's a day-to-day thing. There's a social and economic burden along with that. If you're a seller of the kind of foods that have these things, you know, you're at risk for food allergies. I mean, for recalls due to problems with undeclared allergens getting in there. You know, this leads to the increased regulation, Food Allergen Labeling Act, and food safety. And you know, Charlie's keeping you guys up on that, along with some of the newer regulations. All that sort of, I think, falls into the same things that the council may be looking at when they talk about the big issues that are coming towards the industry. So a little bit more background on what food allergy is. Food allergy is an adverse immune response. That's called type 1 hypersensitivity. It's usually mediated by immunoglobulin E or IgE. It's a special kind of antibody. IgE normally functions to protect us from parasitic or other type infections. And in the case of a food allergy, it just sees some portion of that food as a threat. And it says, hey, you're not supposed to be here. And it starts a cascade reaction that ends up causing itching, rash, gastrointestinal symptoms, vomiting, nausea, and in some severe cases, anaphylaxis. Basically, IgE antibodies on mast cells or basophils, when they get cross-linked after binding an allergen, leads to a signaling cascade and the release of histamine granules. And that process, degranulation, causes the inflammatory mediators to be released and cause people to have, hives, itching, and some of these other symptoms. A lot of work has been done to figure out what. So specifically, nut allergy is one of the most common types of allergies in food. As I said before, they're rarely outgrown. The only accepted treatment right now is strict avoidance. Many of the kids who have this type of allergies carry around an EpiPen filled with epinephrine or adrenaline, and that's in case of emergency. If they have an accidental exposure, their only real recourse is to inject themselves with epinephrine as a countermeasure at this point. I think I summarized most of that already. And again, this is just something to be aware of. This isn't to try to point the finger. This thing is out there. It's happening, and it's just something to be aware of. A lot of work has been done to characterize what makes these foods allergenic. The short answer is there's a small subset of proteins that seem to be the major cause of these allergies. This picture right here is actually a little model of one of the common allergens. It's actually a model I've based off of a pecan, or a model of a pecan protein I based off of a Brazil nut protein. And they are proteins or glycoproteins. And in the case of nuts, they're seed storage proteins. So they're within a seed. They're what give the protein at least part of its protein content and nutrition. They have similar protein sequences and structures. I'll talk a little bit more about that in the next slide. These proteins are they're considered tough. They can survive normal processing, whether you boil them, roast them, pan fry them. They survive digestive, intestinal tract. They resist the digestive of trypsin and pepsin and the enzymes in our stomachs. This table summarizes a great deal of work. These are conserved nut allergens. These are recognized by the World Health Organization and the International Union of Immunologic Societies. These three proteins, 2S-albumin, 7S-bisylin, and 11S legumins, are conserved within nuts. They're found in all nuts. The number and letters basically indicate what sort of size particle they form. So these would be smaller particles, medium and bigger particles. But what's important is that you see this protein in almost all the nuts we've been able to look for so far. And I just highlight here pecan. These two, CAR-I1 and CAR-I4, work from researchers at the University of Florida State have characterized these, at least initially. They also have submitted sequence for this one, although it hasn't been fully characterized. And I want to continue to work on these things and better characterize them. There are others that we have to look for yet, but these three types seem to be conserved. The other thing to point out is that in some nuts, these proteins can be a higher or lower percentage of the nut protein. For example, in the cashew, this NO2 is actually roughly 50% of the protein in the nut, so it's a large portion of the protein in pecan. In pecans, it's more evenly distributed. But in pecans, it's maybe 10% dry weight protein. And of that, only, say 10 or 20% of it may be the allergens. So it's not much in those nuts that's causing the problem. And I think there are ways that we can, either through processing or maybe even breeding, find ways to reduce or eliminate some of these proteins. So my pecan research objectives, to sort of shift gear now, are again to characterize at a molecular level the gene expression, protein accumulation that occurs during pecan development. There have been studies from the past that look at this at an anatomical and tissue level, but I want to look sort of at a molecular level and figure out what you need and how you put them together to build a pecan nut. Again, characterize the pecan nut allergens and hopefully look for develop new processing methods that can eliminate or reduce the pecan allergens. And then I have a little bit of background in diagnostic tools, and so that's another thing that we'll try to work towards, but that'll be further down the road. So what we're doing is I'd like to be able to develop a timeline of the gene expression that occurs as pecans start to develop and then finish. And we've taken a subset of that initially here, and I'll show you that next. Look at the gene expression, look at the protein accumulation, use that to form a basis for further studies to look at differences in cultivars, agricultural practices, generate correlation between the nutritional and sensory qualities of nuts, and just use that information to better guide improvements in pecan growing. So with Charlie's help, we basically were able to grab and collect samples from desirable and summer cultivars for a collection of RNA and protein for analysis. We started a larger scale project to sequence all the RNA that's expressed within the developing kernel site of the pecan from water stage through kernel development. We had to do a couple rounds of RNA extraction, but we finally got everything we need to at least get the process going and sequence a few time points within that process for RNA. I just got the okay on that this week, so we're going to move ahead with that. And I don't have a full story on that yet, but I just want to give you some information of what we're building towards. And then we have some new data alongside the study for gene expression. We're trying to look at the proteins that accumulate. And we're doing that in mass spectrometry and purification of certain proteins to characterize what proteins accumulate in the nut as it develops. And so here are our samples. So as I said, we have desirable and summer from these dates: water stage, gel stage, dose stage, and then full kernel for maturation. And basically what we're doing is we're limiting our sample collection to what's in the void here at water stage within the site where the kernel will develop. So we're taking that material and anything that's sort of adhered to the inside. And we're using that as our sample for now. I know that's a simplified sample, but it's sort of where we need to start to be able to sift through what's actually at the site of the nut development. So one of the first things we did was, because part of my focus is the allergens, is to look at the gene expression for those three allergens, those three conserved allergens I showed you at the table. And what we found was a little bit surprising to me. The gene expression for all three of these allergens during the growing process builds up through early September and then drops off significantly as the nuts mature. And I'm interested in the regulatory process and how that occurs and what influences things like the agricultural practices, watering, and possibly even infection from some of the pests, how that might affect this gene expression, not just of allergens, but of all the other things that are required for building a plant. we saw the same thing whether we looked at summer or desirable. Same sort of trends were there. Gene expression builds up through September and then fell off as the months were short. At least for most of the genes we looked at, not all. One of the questions we have to build on from this is that we can use these now as an internal sort of standard, and we can compare this to the other genes that may be required for the different processes that need to occur, and so we can use this information to help us guide further studies. The other thing we did was to take protein samples and extract protein from the same set of samples. And what you can see is that early on, there's really not much protein there, at least not that we can see when we try to visualize the protein on a gel like this. The first few time points, there's almost nothing there, and then it builds up slowly. One thing to point out from this is if you look at if you remember the RNA peaked at September 12th. That was where the peak RNA levels occurred. But we're really not seeing that much protein accumulate in those samples. Now, we don't have a whole lot of repetitions of this, so this may even out if we look at a few more examples. But it is an interesting result, and I'd like to know what is occurring to drive the RNA down, but yet allow the protein to accumulate. Gene expression, you have a transcription to get your mRNA. So that's got to be the first step, and then you translate that mRNA into protein. So without the mRNA, it's hard to make the protein. So this is a bit of a little bit hard to understand how the protein drops the RNA drops as the protein accumulates. That's one of the things we have to go back and look at a little closer, try to figure out what's going on there. A lot of you have a great deal more experience growing pecans than I do. I venture to say everyone in the room has more experience growing pecans than I do. And if you have any questions or comments about this that might help guide the study or point out certain things that may be obvious to you that aren't to me, please let me know, either before, during, or after the talk. I'd greatly appreciate any feedback. Basically, what we're doing now is trying to go in, as I said, we're sequencing the RNA. We're going to increase the amount of sequence that is available for pecan substantially from this study. Right now, there's only a few sequenced pecan morphs that, at least that I'm aware of, that are in the national database. And hopefully, with this sequencing, we'll be able to get quite a bit more information from doing that study. But we're also going to look at the proteins that we find, and that's sort of the next step here. We're taking these samples and analyzing them in such a way that we can try to pick out and match the proteins that we find to, at least for now, other proteins that are in the database. And again, this is the initial sort of preliminary study, but what we can see is that we're getting a significant number of proteins. I think there's more that we'll find, but we're seeing some of the allergens. I know we're not done because we don't see all the allergens yet, but we know they're there. So we have more work to do. We see structural proteins, housekeeping proteins, are things that are just there to sort of drive the cellular machinery. Things that are going to be there all the time. They're just there to do their job. They're building things, energizing things. There's the things that we don't have a sequence match for, and that's a significant portion of the results that we're getting. So we need to find out what those proteins are, and hopefully the combination of the sequencing and the protein matching will allow us to identify more of the proteins that we're finding. Significant number of the proteins that we're finding are metabolic, meaning that they're involved in the normal cellular processes that you might have a cell, energy generation, signaling, all the things that have to occur just for any cell to survive and grow. Photosynthetic proteins, plant protective proteins, Ripening proteins, signaling proteins, and transcription translation. These are also could be considered housekeeping proteins. This 7% signaling, the 4% signaling proteins, these are the sorts of things that I'm very interested in. There's actually just a paper I found last week describing some of the signaling networks in Arabidopsis and a little plant, wild plant system. And some of the things that they find, I think, will be applicable to pecan development. And so hopefully we can use that information to guide this study and sort of narrow down where we're looking to identify the critical genes and signaling networks that are important for this process. This is just to say that I think what I'd like to see here is 100% overlap, because that will tell me that I think we're doing a good job in our sample collection and our analysis. We may not get that, but right now, from our desirable samples and our summer samples, this area in the middle here is how much overlaps between the proteins that we're finding. We find, in those two sets of samples, we find about the majority of them in both samples. And I think, from what I understand, is they're similar enough that we should find the same proteins in both of those samples, all of them being at least close enough that they would be considered similar. And so I think this just says, one, we need to find more proteins, and two, we need to make sure that we get all those proteins from both samples. So we're continuing that work. And again, I know a lot of this isn't directly applicable to what you guys are concerned with at this point, but I think if we continue this work, we'll find ways to use this information and help the growers. Just to summarize the allergen expression data, we've identified or other researchers have identified the three well-characterized pecan genes, CARI1, CARI4, and 7S-bisphylline homolog. These are tough proteins. They're seed storage proteins. They're there to provide energy and nutrition for the developing embryo and protect it. The allergen levels seem to increase during development through September, and then there's a sharp drop off in October as they mature. We're not sure what causes that, but it's something that I'm interested in. It might point to ways that we could actually reduce, or it might lead to ways that we may be able to reduce these allergens, the expression of these allergens in a small subset of the trees that are grown to produce nuts, specifically for reduced allergen nuts. The other thing to note from this so far is that the protein accumulation seems to lag the mRNA development. mRNA has to come before protein. You see a peak in mRNA expression or accumulation, but it doesn't necessarily coincide with the protein accumulation. And I think there's something to look at there and figure out why that is. And again, I want to expand this work to characterize more of the gene expression, the protein accumulation, and the signaling that's required to build the content. Charlie mentioned we got a little bit of money. We got about half of what I had asked for. So we're going ahead with what we got, but I think as we get these results, we'll try to get that published. Hopefully those publications will allow us to drive and further advance the proposals that we can put forward. And again, any collaborators that we can find who can help us with this process, we're open to that sort of thing. I want this to be get the information out there, and hopefully people can use it. So again, continuing the pecan research, development, build a parts list, continue to learn about the pecan allergens, and as I said, part of my research responsibility is to find ways to reduce or eliminate these allergens, and this will lead to hopefully improve food safety and more people that can eat nuts, including pecans. And thank you. I want to thank the Louisiana Pecan Growers Association. They're helping us out with this study. Dr. Charlie Graham, of course, and several USDA scientists. Funding for the project has come from USDA and the Louisiana Department of Agriculture and Forestry. And I'll take any questions.